It's my pleasure to welcome you to the CEGE CONNEXT Lunchtime Seminar Series. There's a lot of amazing and creative research that's happening in the Civil, Environmental, and Geodetic Engineering department right now. And the value and the impact of this work is only fully realized when it's informing practice and policy. So this seminar opportunity allows you to hear from faculty their perspective on recent innovations that are relevant to practitioners. And we're happy to reconnect with you here today and to highlight that the CEGE department continues to be a resource for you after you graduate. Today, I introduce Dr. Natalie Hull. She is an environmental microbiologist who's an interdisciplinary expert in water disinfection and the relationships between water quality characteristics and the composition of bacteria and other microorganisms in the municipal water supply systems. She also holds an appointment as a core member of the Sustainability Institute at Ohio State, and she is active professionally to support the transfer of research discovery into practice through her leadership and contributions to developing standards for the deployment of low energy LED lamps to be used for UV water disinfection. As a matter of kind of housekeeping, if you will, I ask that you please drop questions in the chat function at any time during the presentation. I will moderate those in the question and answer session at the end of Dr. Hull's presentation. Let us welcome Dr. Hull.
Thank you. Excuse me. Thank you, Dr. McKay. And I'm really appreciative of the opportunity to share some of my work, both here at Ohio State and previous to that, and my perspective on how it fits into the broader picture, looking at the role in particular, one of our focus areas of ultraviolet light in the future of providing safe water for all. So to set up the motivation for talking about UV light, I want to share some of our work that I and others have been learning about our water treatment and distribution systems as a result of our past management strategies and ongoing management strategies. So chlorine, as many people know, it's commonly used for managing microbiological water quality to disinfect microbes at the water treatment plant. And in the US, if you are treating surface water, then the water treatment plant is required to maintain a chlorine residual in the distribution system to ensure microbiological water quality. However, whenever we use any disinfectant or any water treatment technology, you're always balancing drawbacks. So some of the drawbacks for using chlorine are that if we don't achieve adequate disinfection, so on the left side of this, if we have inadequate disinfection, you might have taste and odor problems that develop, or you may have biofilms that develop, or you may have proliferation of pathogens and potentially opportunistic pathogens in your distribution system that can be harmful for humans. On the other hand, if you do achieve adequate disinfection, but there might be other issues that occur. So within the distribution system, you may run out of that chlorine residual, so it gets depleted. Additionally, it requires handling and trucking in and constant supply of chemicals. And you're also balancing disinfection, this first D, with formation of harmful disinfection byproducts that are known to cause cancer and some other toxicity impacts. Additionally, chlorine treatment and distribution issues in some places can select for potential opportunistic pathogens. And we looked at a particular case of this in New Orleans, Louisiana, where they have a unique distribution system. Some of their piped water system is above ground, making it warm and especially amenable to growth of these potential opportunistic pathogens. And we looked from the source water in the Mississippi River through treatment at 2 drinking water treatment plants all throughout the distribution system. And what we found was that some of these opportunistic pathogens, like Legionella, Mycobacterium, and Pseudomonas, these genera of microorganisms that can contain opportunistic pathogens, we found they were at really low relative abundance in raw river water. And then after drinking water treatment, even though the overall microbiological load in those samples was lower, the relative abundance of some of these opportunistic pathogens increased, and that became even more problematic out in the distribution system at TAPS. So this points to the fact that our treatment and distribution management approaches can put people at risk from some of these opportunistic pathogens in the water distribution system. So that was in New Orleans, Louisiana, but we wanted to see more broadly if that was a problem. So we looked at a bunch of medium-sized municipalities throughout the greater Ohio River watershed basin, and we found, again, high relative abundance of some of these potentially opportunistic containing genera of microorganisms like Mycobacterium, Streptococcus, Legionella, and Pseudomonas. So it's not just a problem in New Orleans, it's widespread throughout the US in these chlorinated water treatment systems. And this becomes particularly troubling because people are using water not just for drinking, which we would need to be handling pathogens that cause gut issues. These opportunistic pathogens that we're talking about, Pseudomonas, Mycobacterium, Legionella, they're a problem because they're actually respiratory infections primarily. Pseudomonas can also infect your skin, but people are using water not just for ingestion. They also put it in their humidifiers. You breathe it in when you shower. And so we were finding these opportunistic pathogens in people's humidifiers as well. So this presents a route of exposure to potentially harmful opportunistic bacteria. And it's not just what we're finding. If you look at the burden of infectious diseases caused by two of these opportunistic pathogens, Mycobacteria that cause non-tuberculous mycobacteria infection and Legionella that cause Legionnaire's disease, you can see that they lead into the millions and billions of dollars in combined emergency department and hospitalization costs, and a lot of hospitalizations and deaths. So these are a really big deal for us to manage, and our current treatment practices obviously aren't doing the job because we have these huge health impacts. So instead of looking to chlorine for managing microbiological water quality, to be able to make sure that our microbiomes, the whole microbial community in our water samples, is safe for all types of exposures for all humans everywhere, we need to more holistically manage all the way from the water source through sustainably optimized multi-barrier treatment that can include chlorine as well as other technologies. And we need to have distribution systems that respond to changes in water quality so that we can make sure it stays safe. So we focus on UV in my lab as a way to additionally manage microbial water quality. It has the same drawbacks of any disinfectant of if you achieve inadequate disinfection, but it has some unique drawbacks if you apply enough UV to achieve adequate disinfection. Some of these drawbacks are that historically UV has relied on fragile and toxic mercury lamps. Mercury, if these lamps break and it gets released into the environment, mercury is toxic. Also, UV is just light. It shines through the water. It does not stay in the water. And so there's no residual disinfectant that gets maintained to make sure the water is safe after we treat it. And then finally, it requires a lot of electricity. So we're focusing on ways that we can overcome these strategies to help implement UV more, because UV is almost a perfect partner to chlorine disinfection. If you look at the sensitivity of different types of infectious agents to chlorine, the things that are most resistant to chlorine, like protozoan oocysts, those are most easily disinfected by UV. And on the other hand, the things that are most resistant to UV, which are viruses, they're the most easily inactivated by chlorine. So they create a perfect partner for managing microbial water quality, and they can also help us minimize some of these drawbacks. So just to remind you, if you're rusty on your physics, UV is just a high energy part of the electromagnetic spectrum, just below Roy G. Biv in the violet visible light region. And as we decrease wavelengths, we increase energy. So we're focusing on this region of the UV spectrum that has really high energy, and it's able to disinfect microorganisms. Mercury-based UV disinfection for water systems has a proven history of disinfection. It started with applying low-pressure mercury lamps, which emit approximately one wavelength of UV at 254 nanometers, and it's been applied for almost 100 years. And so we have really good literature on how big of a dose of low pressure UV it takes to achieve 4 log or 99.99% disinfection of a wide variety of bacteria, protozoa, and viruses. A little bit after the development of low pressure UV came medium pressure UV, which just has a higher vapor pressure of mercury within the lamp. But what is important about it is that it emits a lot of wavelengths across the UV spectrum. It also emits a lot of wavelengths in other regions, but these UV wavelengths lead to a much lower dose required to achieve the same level of disinfection than we previously saw for that single wavelength. So this began to tell us that multiple wavelengths make it easier to disinfect microbes. And so people started studying this in depth, trying to look at the dose required to achieve 90% disinfection or 1 log across all the UV wavelengths for a variety of different infectious agents and indicators. And what you can see is that across the wavelengths, in some wavelengths it requires a low dose, and some wavelengths it requires a high dose to achieve disinfection. And so this tells us where we can target, where the microbes or viruses are most sensitive to UV, because it takes the lowest dose to disinfect them in these regions. So it's going to make it easier for our UV light to achieve the same level of action. And we're starting to understand the reason that microbes have different sensitivity across the wavelengths, especially for things like viruses. I'm showing here an MS2 virus, which is an RNA virus, and an adenovirus, which is a DNA damage virus. We're starting to see that damage to these molecules that make up these viruses varies across the UV spectrum. Historically, because we started off using low pressure UV, which just emits at 254 nanometers, we only understood the impacts at 254 nanometers. And at that wavelength, primarily DNA damage and RNA damage contribute to disinfection. But when you start looking at other wavelengths, you see things like protein damage are really important, especially at low wavelengths. So this not only tells us which wavelengths to target, but why it works better. But we're still looking at mercury-based UV sources when we're thinking about that. But recently, there has been huge development of mercury-free UV sources. So that gets us one step closer to removing these drawbacks for implementing UV more widespread. So some of these mercury-free UV sources that we've been focusing on in my lab include krypton chloride excimer lamps and light-emitting diodes. And they're really exciting because their emission spectra varies across the UV spectrum, but it just so happens we can select them so that they line up with those regions of the UV spectrum where it's most easy to inactivate microbes, where they're most sensitive to UV. So it gives us not only a less toxic, less mercury-based UV approach, but also a more effective approach. So I'm gonna take you through some stories from my lab using LEDs and also the excimer lamp, and then combining them together to show you how we're using these mercury-free UV sources to overcome some of these barriers. So starting with the LEDs, we validated the first commercially available UV LED water treatment reactor across a range of flow rates and water qualities, and we measured disinfection of this virus MS2. And we used something called combined variable modeling to develop this predicted MS2 log inactivation as a function of what we observed based on those different conditions of flow rate and water quality. And we were able to use this model. It kind of tells you the operational performance of your reactor across any conditions. We use this model that we developed at the bench to compare it to what happened when we actually installed it in the field in a rural drinking water system. And so to figure that out, we compare results from whenever we go up there and challenge this with our same MS2 virus to our existing model for this reactor performance. And if the slope of the line is in this region, that means we are getting better disinfection than we predicted for MS2 disinfection. And what we saw over the course of a year of challenge tests is that, in fact, our reactor was at least as good or better in terms of disinfection performance all throughout the year. So this was a successful demonstration that we can already, with really low maintenance, implement some of these new LED technologies. And this was true even though I installed it up there, I didn't clean it, I didn't turn it off, it was operating continuously throughout some pretty challenging conditions for UV disinfection. So this UV reactor was treating water from the slow sand filter at this rural water system, and they were experiencing high turbidity events in their filter effluent during the warmer summer months. They also, in the spring months, whenever there was snow melt in this high alpine water system, they had really low UV transmittance, which is a measure of how clear the water is to our UV light. And so despite these challenging conditions, the reactor still maintained performance. So this was really exciting that UV LEDs are already that much closer to being available for widespread implementation, even in rural small systems. To further investigate those challenging conditions, my master's student, she's also a visiting scholar from TU Delft. She looked at some really high turbidity conditions, turbidities up to 400, 600. These are way higher than you would ever expect to disinfect with UV light. But we wanted to know, worst case scenario, can we rely on UV whenever small rural systems might be experiencing these challenging conditions and they may not have as much monitoring and response capability as some larger utilities? So in this terrible water quality, Judith found that even though turbidity was highly variable and a lot of other water quality parameters were also highly variable, throughout the course of the year with her UV disinfection tests, UV disinfection was highly predictable for these indigenous spores that were already present in the water. So that is also encouraging, that we can still get measurable, predictable UV disinfection even in these challenging conditions. So going back to the study of implementing the LED in the field, we wanted to get a bigger picture of how it is treating the overall microbiomes in those samples. So my PhD student, Yijing, and undergrad, Amanda, they were looking at this from some of the samples of biofilm from the tubing for our UV reactor. And we were comparing it and water samples from the drinking water treatment plant filter influent, slow sand filter effluent, chlorinated water. So this is equivalent to the drinking water for this rural water system versus our UV LED effluent. So in the water samples, each point represents the overall microbiome for a sample collected at some point throughout the year. We saw that all the samples were overlapping. There's really no significant differences in our brief snapshot water samples. However, if you look at the biofilm samples, you can see that the chlorine samples are clustering off to the side. They're way different than the UV LED samples, which are overlapping with our filter effluent samples. So this is telling us chlorine is doing something to shift that microbial community to be different from that filter effluent. And this is important because biofilms capture more of what is going on long-term with the water. So these water samples were just one day that went up to collect a sample, but biofilms were allowed to accumulate for anywhere from 3 months to a year. So they give you a more integrated picture of what's going on. Another reason this is important is because of the types of microorganisms that were significantly different in this shift. So in those chlorine samples, those biofilms, we saw greater relative abundance of some troubling genera like Streptococcus, Staphylococcus, Pseudomonas, Escherichia shigella, and Acinetobacter. So a lot of enrichment of some of these genera that can contain bacteria that could cause a problem under the wrong circumstances. We're still troubled by the fact that UV LED was enriched in Mycobacterium, because as I was showing in some of the other plots, Mycobacterium is a major problem even in chlorinated distribution systems throughout the US. So focusing on these mycobacterium is something that we are continuing to do. We recently got a WRF grant to look at this. And it's really important because even though mycobacteria disinfection has been studied along with disinfection of other pathogens and indicator bacteria, mycobacterium disinfection hasn't been studied for any of these mercury-free UV sources. And it's troubling because, as you can see, it takes a higher UV dose to get the same level of inactivation for mycobacteria. So this is a really important thing to figure out that we're going to be focusing on. And I think it's important to figure out how all water treatments impact mycobacteria, not just UV. So those are the LED stories I'll share with you. Now I'll get into some excimer lamp studies. So we're looking at this low wavelength lamp emission. The first study I want to share with you is about comparing 222 nanometers, so that excimer lamp emission, versus 254 nanometers, which is commonly emitted by the low pressure mercury lamp. Because another reason this is exciting is that for exposures, for example, not in water, but maybe in air, or maybe to manage airborne risks at some place like a wastewater treatment plant where there may be risks from aerosolized microbes in the wastewater to the workers. Implementing UV 222 to help keep the air free of pathogens is exciting because it is safer for human exposure. If you look at the human exposure limit for low wavelengths like 220, the threshold limit value, TLV, back in 2020, it was 25, but they've actually recently increased it to 600 something because the more studies that come out, the more they realize that it is more safe than we initially thought for human exposure. And that's different than 254 nanometer, which causes skin cancer and eye problems. And so 222 is safer for human exposure. And what's really exciting is that we were looking during the peak pandemic at the impacts of 222 on SARS-CoV-2 as a way to prevent transmission of viral airborne pathogens. Whenever we looked at disinfection of SARS-CoV-2, we used standardized procedures to calculate UV dose, which a lot of air research is having trouble with, but we did it the best we could working in the biosafety level 3 labs. But what we found is that the dose required to achieve 1 log of disinfection of SARS-CoV-2 was equal to 1.6 millijoules per centimeter squared for UV 222. We didn't do the 254 nanometer study with SARS-CoV-2, but if you look, this is a reminder of that literature I showed you before. If you compare this to the dose required to achieve a log of disinfection of the viruses, all the viruses in the literature for UV, you can see 1.6 is way lower. It's on the outlier side of the spectrum. SARS-CoV-2 is super sensitive compared to other viruses, especially compared to how you can disinfect them with 254. So we were excited to see this. Another thing that we looked at for disinfection of bacteria in water with comparing 222 nanometer light from the excimer lamp to 254 nanometer light emitted by the traditional mercury lamps, we focused on an antibiotic resistant bacteria, Bacillus subtilis. And whenever we expose Bacillus subtilis to different doses of UV 222 in red or UV 254 in blue, we saw that for a given UV dose, we got more disinfection of Bacillus subtilis. So 222 nanometer is better at disinfecting this bacteria than mercury-based low pressure UV light. Not only that, but if we look at the actual genes that contribute to antibiotic resistance in this bacteria, we can look at their damage as a function of UV dose in 2 different types of experiments we did, where we used whole cells, or we first extracted their DNA and just looked at the damage to the genes alone. If you look at these in summary, for a given amplicon length, that is what the end numbers here are on each of these entries in the legend. and everything for the excimer lamp is in shades of red, everything for the low pressure lamp is in shades of blue, you can see for a given amplicon length, the damage to the antibiotic resistant gene was higher by the excimer lamp than it was to the low pressure lamp, or at least very close in most scenarios. So it was similar damage. But what this translates to is more important, because just having the gene damaged doesn't really cause much of an impact when we're worried about these antibiotic resistant bacteria spreading or sharing their mechanisms of resistance. So what was even better was that the excimer lamp drastically was much better at preventing cells from sharing those resistance genes through this mechanism of horizontal gene transfer. So this is super exciting that not only does it disinfect better, but it also prevents those cells from sharing their harmful antibiotic resistance genes with bacteria that don't have that resistance. Another study using the excimer lamp is addressing one of the problems with UV disinfection related to the fact that there's no residual to maintain microbial water quality. And that's the fact that some microbes can repair themselves after UV disinfection. They can repair their damaged genes. And so we wanted to look at the impact of different wavelengths on reducing that repair and subsequent regrowth. So David, undergraduate, he compared disinfection of E. coli by the low pressure lamp emitting 254 in blue and the krypton chloride excimer lamp emitting 222 in red, and they had similar disinfection kinetics. But whenever you look at the regrowth of the E. coli, that permitted conditions where the cells could repair themselves after UV, we saw that the concentration of cells after 254 treatment, they in fact did repair themselves, and their concentration increased as a function of photorepair fluence. You can think of it as a time. On the other hand, those cells that were exposed to 222 nanometer UV, their concentration decreased, so they continued to die off. They did not repair themselves. They got even more sick and died. So this 222 nanometer light allows us to combat one of those problems of not having a disinfectant residual, because it reduces that mechanism that cells can repair themselves and regrow. One more exciting study with 222 nanometer is looking not at a microbe itself, but at one of the molecules that microbes produce that is harmful. So it's a common problem in Ohio and the Midwest, in the Great Lakes region and throughout the world, that we have harmful algal blooms that produce algal toxins, and cyanobacteria do this. One of the cyanotoxins that we were looking at was microcystin LR. And so we were comparing degradation of this hepatotoxic chemical by our two different wavelengths here, 222 in red and 254 in blue. And if you look at the rate constant for degradation, you can see that it is much higher for 222 nanometer UV. That means UV 222 is much more effective at degrading the microcystin LR for a given UV dose. And what is the variability captured in these error bars here represents a range of water quality conditions where we had natural waters from the Ohio region that had really low water quality, especially at these low wavelengths where UV 222 has high absorbance. It also represents a variety of different measurement techniques, measuring different properties of the toxin. And we did some of those in conjunction with USGS. And we recently got a grant to continue looking at this from the HABRI. And we're really excited to continue that for more toxins and more cyanobacteria. And the reason it's so exciting is because of what just popped up on this secondary y-axis here, is that even though the excimer lamp, this 222 nanometer lamps, they're still in really early stages of development. Low pressure lamps, they've been around for 100 years or more, and they have them dialed in terms of electrical efficiency and operation. And water is generally more clear at those higher wavelengths. if you take into account all of those drawbacks and compare across 222 and 254 to calculate how much energy it requires, even though the water is less clear and the lamps are less efficient for UV 222, you can see in the diamonds the electricity that it takes to achieve the same level of degradation is comparable. So UV 222 is so much more effective. It is electrically competitive, even though it hasn't been electrically optimized yet. So this is super exciting. With some slight optimization, it's going to be even better in no time. So those were studies focused on the excimer lamp. Now I want to share with you about some of our studies looking at light emitting diodes combined with the excimer lamp. So we can achieve this multi-wavelength strategy from putting things together to get different types of advantages. So you can combine UV LEDs or other UV sources alone. So you expose your sample to the entire dose, for example, of a 255 nanometer LED. And if you do that and look at disinfection on the y-axis as a function of UV dose on the x-axis for 255, 265, 285 nanometer LEDs, or the 222 nanometer XC lamp, or the low pressure lamp, 254 nanometers, alone, you can see that the excimer lamp is most effective based on dose response. And you can compare that to the commonly used historically low pressure lamp here. It is a little bit less effective based on dose response. However, a lot of people are using these 285 nanometer LEDs. That's what was actually used in that implementation study, the year-long study that I shared with you about, because you can get the most output for these LEDs at the higher wavelength and have high electrical efficiency. So there's a lot of competing variables that go into selecting what goes into this multi-wavelength strategy. So we investigated part of that by applying UV sources in sequence. So we would take a sample and expose it to half the total dose from one UV source, and then expose it to the second half of the total dose from a second UV source. And here again, I'm showing you on the y-axis the energy that we calculated that was required to achieve a certain level of disinfection of our microbe we're focusing on here, MS2 virus. And you can see, because low pressure lamps have been optimized for so long, they have the lowest energy required whenever you apply them alone for disinfection. Similar to where we had competitive electrical efficiency between low pressure and the excimer lamp for degradation of microcystin LR algal toxin, we see competitive electrical efficiency between the excimer lamp and the mercury lamp for MS2 disinfection. And then our LEDs, they are not quite as efficient yet and less effective for a given dose. So we can see they require roughly an order of magnitude more energy to achieve the same level of disinfection with our current technology. So that was for the single exposures. For those sequential exposures where we apply them in a certain order, we can see that overall the electrical energy required tends lower. And what's really exciting is that all of these are competitive with current electrical efficiencies for that multi-wavelength mercury-based medium pressure UV lamps that we have. So already we could implement these novel UV sources of excimer lamps and LEDs, or combine them with existing low pressure lamps to achieve better disinfection with competitive electrical energy requirements. So those are all the ways we've been looking into applying mercury-free UV in ways that will better or apply more help in managing microbial water quality. Though we still have these drawbacks for UV of not having a residual in the distribution system and that they require electricity. So to get around this no residual drawback, we've been looking at the feasibility of applying UV throughout distribution systems to achieve this. And so we put together our concept of what this might look like. So for municipal water systems, if you apply whatever suite of treatment technologies at the water treatment plant, and that could include UV or not, we think that there is possibility for applying UV in large diameter transmission mains, storage tanks, distribution mains, or in point of entry whole house UV systems, or point of use under sink UV systems. In non-municipal systems, we think you could also use point of use UV devices or point of use and point of entry UV devices in decentralized settings or for well water to achieve good microbial water quality and protect public health for a variety of uses throughout these systems. And this will give you a boost of disinfection power wherever you apply them. But trying to figure out where to apply them is a problem that needs some attention. So we looked at that and we developed a risk framework that can be used to help figure out, okay, how frequently do we really need to put these UV booster stations? So PhD student Dan, he looked at a scenario reflecting an off-grid decentralized rural water system that might be in developing communities where people collect water from a community access water point and they transport it over some amount of time to their house. And during that time, we may have those conditions where cells that were initially UV disinfected could possibly repair themselves, like I told you about with David's study. And so Dan was looking at a variety of factors that could contribute to the risk from drinking water that might have a reactivated microbe in it after storage and transport after UV disinfection. And so what he found was that if you look at the risk of infection across a variety of collection times, so this is the time that people visited the water access point and got their UV treated water, versus exposure time, this reflects how long it took them to walk home or how long it takes the water to sit out in storage before they consume it or are exposed to it, as a function of different contamination levels in the source water. And so we see that at low contamination levels, the risk of infection never exceeds this commonly accepted value for acceptable risk of 1 × 10⁻⁴, or fewer than 1 in 10,000 people get infection from that exposure. We do see at high contamination levels, there are certain times of exposure time and collection times that line up with higher risk periods. So to prevent risk from those events occurring, you can select travel times or between UV treated water access points that make sure people don't get into the red zone. You make sure that no one has to travel the amount of time that would put them in this risky scenario. So if we have a rural place, you can figure out where to put your community water kiosks that are treated by UV, such that they're always in the blue zone. They're not going to exceed that risk. No one is going to have to be in that danger zone. So that paper was the framework of these decentralized developing community areas, but it can also be applied for more municipal types of applications as well. So similarly, we could use that risk-based framework to figure out the water travel time to determine how frequently we need to add these UV booster stations. It's also applicable for some of the types of municipal system management that is being encouraged lately in the US by the EPA, and that is for rural areas that are currently not served by a municipal water system, or maybe they're served by a failing water system. There's been a lot of talk about extending pipes to serve these underserved areas, or regionalizing water networks so that they can pool resources. But adding longer pipes means more opportunity and more travel time, more potential regrowth of microbes, more potential for cracks and leaks to let dangerous microbes in. So you can use this risk-based framework to figure out how many UV booster stations would it take to keep the water safe through that entire travel time to serve this previously unserved set of rural houses. Okay, so that is how we're conceptualizing applying UV throughout distribution. But we know that electricity and water don't really mix. And we're not going to put power the grid all throughout our underground buried water pipes. And so we need to figure out a way to get around this barrier of the electricity requirement. And so we've been looking into coupling UV with hydropower throughout the distribution system in contained units so that the power is generated right where it gets used by the UV system. And Dan, PhD student, and undergrad Jake have been working on this primarily. So they have been comparing whenever we test different UV systems, starting with this 3B system, powering it by the traditional way that you plug it into the wall versus hydropower, the technology they've been developing, versus a hydropower battery technology that we've been looking into that would be more applicable for an intermittent type of supply. And they compared disinfection of MS2 by this 3B reactor as a function of flow rate in the triangles versus the MS2 concentration with the UV off for those flow rate conditions. And what we see so far is that plugging it into the wall gives us the disinfection we expect. This is what the company designed it to achieve. And our hydropower battery system also aligns with that disinfection. But so far, our hydropower system, the purple, those haven't been giving us the same results. And that's true whether we test this small 3B unit or test some slightly larger UV units. We see the same trend where our hydropower systems aren't powerful enough yet. So we looked into why, and we calculated the efficiency of our current, you can barely see it here, our hydropower turbine plus UV LED setup. And we calculated the actual efficiency of this super cheap off-the-shelf turbine that we bought on Amazon. And we found that we're getting currently about 1% efficiency by dividing the actual power we measure by the theoretical power that our turbine should be able to achieve based on pressure head loss. If you look at the required power for these three different LEDs as a function of flow rate based on theoretical power at those flow rates, you can see we need up to about 25% and down to about 5 or 2% for the 3B, and we need higher efficiencies for the larger UV unit. But what is important here is that we think a modest increase in efficiency that we need to be able to give our LEDs enough power to get in this dark purple required efficiency, we think it's achievable soon because there are already in-pipe hydropower systems that have around 50% efficiency, and hydropower in general is a really efficient technology. Dams, for example, are more than 90% efficient, but we just haven't got that economy of scale in this pico hydropower scale application we're working with. But we've recruited some help. Dr. Clarissa Bologna here at Ohio State, she's a hydropower expert. She's joined up with us, and we've recruited an MAE capstone this academic year to be working on optimizing a turbine to get us that jump that we need to adequately power our UV LEDs. On the other hand, as UV LED efficiency increases, that will also decrease this required efficiency. So both of those factors together, increasing our turbine efficiency and decreasing or increasing the LED efficiency, both of those will get us closer to achieving this goal of using hydropowered LEDs to get us away from those boundaries for applying UV disinfection more broadly. So those are the boundaries that we have been addressing for applying UV disinfection to help manage microbial water quality and keep it safe from a variety of microbial concerns, whether it's opportunistic pathogens, toxins, or regrowing bacteria. But there's one boundary for doing this that I wanted to circle back and tie it in with the beginning, and that is our regulatory framework in the United States. So other countries, they don't have that same requirement for a chlorine residual in surface water treatment municipal systems. They instead focus on producing water that has really good biostability so that it maintains microbial water quality throughout their systems. And so the fact that we have this regulation focusing solely on chlorine as the way to maintain that microbial water quality is a big roadblock for achieving our concept of being able to use UV instead or as a complement to achieve this goal and protect public health for all. Thanks again for your attention, and I look forward to your questions, and I could go off on any of these topics for a long time, so feel free to ask anything.
Thank you, Natalie. And yeah, I'll invite folks, if you want to put a question in the chat, feel free to do so. We're also a reasonable sized group today that using the raise hand function is also another way to be able to share your ideas and learn a little bit more. Right. Well, it seems that there's not a lot of questions today. So I want to thank you, Natalie, for being with us and really framing together a very comprehensive viewpoint of how your work intersects in many different ways and addresses quite some big challenges in terms of maintaining water quality from a biological perspective, that safety associated with it.
Yeah, thanks again for the opportunity to share. And if anybody has questions, feel free to email me. I didn't put my email on this slide, but it's on the website for the CONNEXT registration.
Thank you. So just as follow-up then, those of you who requested the CPD hours, those will be available in about 1 to 2 weeks. That we'll send them out by email to acknowledge your participation here today. And I invite you to join us again in our next CONNEXT presentation will be in mid-April. And we'll kind of look from the geodetic lens from the Civil Environmental and Geodetic Engineering Department. So thank you all for joining us today.